All right, so I'm presenting some work that has recently been published in a paper that the first author is Roberto Tejero from Valencia, Spain. And he is shown here, and it's we are using AlphaFold to model small proteins and to see how they compare with the NMR data. And the other authors are Janet Wong, and I also helped, and I'm in the lab of Dr. Montelione here at RPI. So one thing we all heard about AlphaFold and we wanted to try it out. So we had a resource back from the NESG of 40 NMR X-ray pairs with NMR data, and decided to use these to evaluate how the AlphaFold structures, see if we could evaluate the AlphaFold structures. All right, so our initial goal was to evaluate the accuracy of some AlphaFold models. And as you've heard from the other talks, like these AlphaFold structures are very good, and which is also what we learned. So we specifically decided to start with 6 proteins that had NMR X-ray pairs and good NMR data. And we used AlphaFold Multimer with no homologous templates to calculate the structures. And so as Jonas Adler pointed out, the templates, having templates didn't make a huge difference in the AlphaFold structures, but we wanted to make sure that we weren't using the actual, X-ray structures that were in the PDB, like when we use the AlphaFold to calculate the structures. So then we have the AlphaFold structures, NMR structures, and X-ray structures, and we're going to evaluate them using structural alignment. This RPF DP scores, which has been developed in the Montelione lab, and to see how well they agree with NMR data, and then also the RDC Q factors. And I very much would like to thank Angus for his introducing RDCs so that I can spend a lot less time on that. So also, thanks to Jonas for talking about the Cas14. I think most people are familiar with it. In Cas14, they have crystal structures that are not in the database yet, and people are using their prediction methods to predict those structures. But in the most recent round, round 19, there were actually 3 NMR structures that were as part of Cas14. So this paper by Janet Wong from our lab, she went back and did an analysis of these 3 structures, these 3 NMR structures, and went back actually with the authors of this structure and ended up doing recalculating a structure because there were some problems with the original structure that Nick Fowler had pointed out. This structure didn't have a complete nosy peak list, and some of the assignments were a little bit wrong. So they ended up recalculating the structure. So for these structures, we don't have an X-ray crystal structure, but we also included them in this paper to do analysis of how the NMR structures compare with the NMR data, and how AlphaFold structures compare with the NMR data. All right. So the first thing we do before we started this analysis is we have to define the regions that we're going to use for like the overlap and for the evaluation. So for the NMR model, we typically would use this software called SciRange or FineCore2 that shows you the ordered residues and the disordered residues. And so the well-defined regions for this NMR structure of 20 on models in the ensemble, we choose residues 305 to 426 are the ordered residues. But then for the AlphaFold 2 model, we calculated an ensemble of 5 structures. So back when we worked on these when the Cas targets were worked on, was when AlphaFold 2 was available. The NMR X-ray pairs, we used the AlphaFold Multimer software, which is the newer one. So for here, we had decided to use the reliably modeled regions with the good PLDDT scores, and also leave out any regions that were not converged. And so then you get the regions that you're going to overlay, and you can overlay them and get your GDT score, which is 0.9, which is very, very good. So we did this for all the targets, and then the next thing to do is to look at the structural comparison. So in this case, you can see the correlation for these small proteins, especially this one, is very, very good. So what we're showing here is for the target, which is called GMR137. That was the NESGID back when it was solved by the Northeast Structural Genomics Consortium. You have along the diagonal, you have the ensemble RMSD showing that, like, for example, the AlphaFold ensemble 0.2 angstrom RMSD is a very tight ensemble. And that has been typically the case that we've seen, that they've AlphaFold, the 5 structures usually have a very tight ensemble. And then on the upper diagonal, we're showing the GDT score. So when it's a very high number, it means that it's they're a very good match between the structures. So 100 would be a perfect match. So you can see here that the AlphaFold structure, which is shown in red, and the X-ray structure shown in gray are almost identical. And then if you want to look down here, this is the C alpha RMSD, and you can see that the X-ray structure and AlphaFold structure have a 0.6 angstrom RMSD. So of course, that's really amazing. Then we also have, for 3 of the 6 NMR X-ray pairs, we have RDC data. And so if it has an asterisk here, it means that the NMR structure was solved using 2 sets of NH RDCs. And so when we do that, then we do this comparison again, you can see that if we look at the GDT score, you can see that the very best agreement with the X-ray structure is the NMR structure that is refined with NH RDCs. And then also the AlphaFold structure is almost as good. And then if you look at RMSD, you can also see that this NMR structure refined with RDCs, 0.78 angstrom, had a very good RMSD with the X-ray structure. So most people know that adding additional restraints and adding the NH RDC data will make your structures better. And this shows that it makes them more accurate. So moving on, we also want to compare the structures to the NMR data. So we do this using this RPF DP score. It's a measure of how well the NOSI data, the NOSI peak list, will match to the structural models. So what you do is you start with your structural models, then you have your NOSI pick lists in your assignments, and you generate a distance network or a graph, which is shown here from the query structure. So you have like your ensemble, you have your assignment. So these green circles represent like each chemical shift assignment. And then you draw a bar between them, you connect them with edges if your distance is less than a distance max. And typically this would be like 5 angstroms, but of course it would depend on how you collected your NOSI data or your system. But we'll just say like less than 5 angstroms. And then you draw, like, you have your graph of what you would expect from these structures. And so then the next thing you do is you also take your NMR data and you make your graph of your ambiguous NOE network. And so now again, all these red circles are your protons from the resonance assignments, and the edges that connect them are all the potential NOEs like from the NOSI peak list. So for example, some NOE might have like several different assignments, and you have a certain match tolerance that you get this ambiguous NOE network. And then you can also show this as a you can show it as a contact plot, like where you have the residue and then you have the context that you would expect to see from the structure. And this is a contact plot, but it would be an ambiguous contact plot. So now what you are going to do is compare these 2 graphs to see how well the structures that you have are matching your NMR data. So if they agree, like they're in both, like you expect a short distance, you see a peak, it will be a true positive, and it will be shown in green. Then if you have a short distance, but you have no peak, then it's called a false positive, and that's shown in red. And then if you have a peak, but it can't be explained by any of the structures, then you have a false negative. And of course, there's like a lot of true negatives, which is when you don't expect a peak, there is no short distance, and you would have a true negative. So this is something that has been developed in the Montelione lab by Janet Wong, and there's some references down here. So what we do is we do this calculation, and then we pull out these metrics from like information retrieval statistics. They're pretty common metrics, and people in AI, like in machine learning, use these a lot. So we have a precision, which is true positive divided by true positive plus the false positives, and the recall, which is the true positive divided by true positive plus false negative. So you can imagine that if you have no false negatives, like no peaks that are not explained by the data, then this would be 1. And if you had no false positives, so no expected peaks that aren't there, this would also be 1. So these are measuring the percentages from the structures whose back calculated NOE cross peaks are part of the original peak list. So so it's basically how good is your structures matching your data. And then also recall, you can measure the percentage of peaks that are consistent with your query structures. And then we also have down here an F measure. It's an overall performance score that's calculated from the recall and precision. And so this is basically like an averaging, like a mean of these terms to give us a metric. So so the very last thing we do is we want to normalize that F score, that metric, to get discriminating power. So what we do is we normalize it so that it runs from 0 to 1. And so 0 would be like any random, you take your protein structure and you make a random chain, so it's not folded up. You would hope that would have a match to the data of 0. And then if you had a perfect completeness of the noisy, perfect completeness, your best performance, and considering the completeness of the noisy spectrum, because sometimes noisy data is not perfect, it's never perfect, and you would look at what you would expect for your short 3-bond connected protons, and you would kind of normalize for the quality of the noisy data, and that a perfect score would be 1. So this is something that we and myself used in the NESG to see how when I was solving NMR structures, like how good is my structure? We really want the F measure to be greater than 0.9. We want the DP discriminating power score to be better than 0.7. So what I'm showing here is for these 3 CASP14 targets. In gray are the CASP14 models, so the predicted models, and here we have GDT, so perfect, and it's GDT2, so it's agreement in structure to the NMR structure shown in red. So to the best, highest DP scoring NMR structure. And so if you have 100, then it means it's perfect agreement. So we show here that DP score correlates pretty well with the GDT to the presumably accurate structure. And then also we have here the AlphaFold structures. So here we had a case where the AlphaFold structures got a higher DP score than the NMR structures, which could mean that they're better, but basically it means that overall they are fitting the NMR data slightly better than the NMR structures itself. So that's actually very interesting and could possibly be used in the future to learn something about our NMR structures. In this case, actually, let's jump over here. In this case, they're pretty similar in DP score, and they're pretty close in GDT. They're probably maybe 1.5 angstroms different. So the interesting one is right here, and I'm going to come back to this at the end, because this is a case where the AlphaFold structure has a lower DP score than the NMR structures, and it's not having the best structural agreement. So perhaps we can use this to learn something about our NMR data and the AlphaFold structures. So I wanted to show this to students that we have an RPF server that can be used to get the RPF recall precision and F score and the DP score. And so it's at the Montelione lab, chemrpia.edu/RPF website. So what you do to use it is you just put in your protein name, whatever you want to call it, and then you choose your chemical shifts and you put them in as a BMRB file. It actually will accept BMRB files in version 2 and 3 format. And I think in the future, like soon, it will probably accept other formats. So we also have, you put your PDB coordinates, your structure here. You can have your ensemble of 20, or you can have one X-ray structure. It's up to you. Then the next step is to put your nosy peaks. So you don't have to, you can put whatever you have. So in this case, it can handle a 2D nosy, a 3D N15 nosy. In this case, I had an N15 nosy, and I had a carbon nosy aromatic and a carbon nosy aliphatic that I collected separately. You could have it collected together, or you could have you can even have a 40 HC nosy, which is and so it would be really nice. We could probably also include that the fancy nosies like for MPRO that Angus talked about, the HMQC, sorry, the 3D nosy TROSY. So any nosy, it can take into account any different nosy peak lists. So then after you you put in your information, you hit submit, then you get to this next page where if you want to include, so your peak list can be in Sparky or XEZ format, and you have your columns. And so it actually could be any format where you have your peak list, where you have the chemical shifts, and then you have an intensity. So what you tell it here is you find the N15 column, and you say that's column 3. And then if you have any folding, you would want to put in the sweep width for folding here for nitrogen. And then for the proton that's attached to this nitrogen, which is called HX1, that is column 4. And you can see that those are HN's chemical shift. And then the indirect proton is column 2. This, Janet has been working on this software, and now it automatically fills in these columns for you, and it automatically puts sweep with a big number, so there's no folding of the peak list. But if you did have folding, you would want to put this in. And so my next slide, I explained a little bit like what I mean by folding for the sweep width. I have an example where I collected a nitrogen HSQC and also in my N15 nosy with folding in the nitrogen dimension. I think it was about 27 ppm. These are the side chain NH epsilons of the tryptophans. And so I folded them so that I could make the sweep width smaller. We often fold we always fold, like, you know, arginines and lysines that are way up at like 85 ppm. We fold them down here. So it's actually really important to know your folding sweep width. And then here's an example where I accidentally chopped my carbon dimension a little too small, and I folded these isoleucine delta peaks from up here, and I folded them down here. So if you don't put in the correct sweep width for folding, your peak list, you know, won't be matching the peaks. So that's an important thing to consider. So after you hit submit, you load it, you get the summary. So the summary that you get is the recall, precision, and F score that I talked about earlier for your query, which is your input. So you have, here we have 0.96, 0.9, and 0.93. So it's a very good quality structure. And also it gives you the final DP score, which is 0.82. And so in NESG, we made sure that we said it wasn't a good match to your data unless it was better than 0.7. Better than 0.8 is even better. So so this is a good quality structure. Then and then it's also normalized by what the scores you would get for like any freely rotating chain, and also this is the score you would get for the ideal structure. And then what is this? And then the M score is actually kind of a measure of like how good your nosy data is, like what percentage of these like two bond and 3 bond like proton interaction peaks that you would expect to see in your nosy data is missing. So this means that only 10% of your expected peaks are missing. So this nosy, for all the nosy data. So this is a good thing. And then the other thing it gives you is that it gives you this molecule. You can rotate it and you can see the residues with precision violations. And so, for example, residue 18 here is isoleucine 18, and it has precision violations. So so what does that mean? So you can actually click on precision violations and recall violations and see them, and you can get information about each of the nosy peak lists. You can also get the DP score for single models by clicking on this, and you can get an average for the single models. It's a little bit lower than the average DP score. So let's see. So when you click on the precision violations and then you can give it a residue, or you can look at all of them, you can give it a distance. So basically we're looking for short distances in the model that are not, that have no nosy peaks. So basically we're missing the peaks. And why are we missing the peaks? So in this example, I said 13A, and I included, and I said less than 3 angstroms, and it came up with these peaks from the carbon nosy aliphatic. And it turns out that also in the N15 nosy, we're missing this peak and so this is the distance. So this is like a 2 angstrom away, but there's no peak. And so why is that? So it turns out if you go back to your data, it's because this H alpha was at 4.79 and it was overlapped with water. And so those peaks were not there. So anytime you have like precision violations, you kind of want to know why, like, why do you have peaks that are missing in your data? And as long as you know why and can explain it, then it's okay. And then the next thing you do is you look at the recall violations. So basically any peaks that can't be explained by the model and resonance assignments. And so you have extra peaks. And so if you have a lot of them, they could be noise, but if they're like high intensity and you look at them and they are there, it could actually mean you're missing some assignments. So you can actually click on these, you can sort by intensity, and you can click on them and see what are the possible assignments. And so this can also help you figure out like what these recall violations are. And so this is something I used a lot when I solved a lot of NMR structures in the NESG. So getting back to the results from AlphaFold, what we have here is the RPFDP scores. So for three of the structures. So we have the NMR structures, and then also we have these Rosetta refined structures, which was a different force field that was restrained with was calculated with restraints. And so the first thing you can see is that when you look at the F scores, like these are all very, very good. They're all above 0.8, they're close to 0.9. You can see these DP scores are also pretty good. We wanted them to be above 0.7. In some cases, they're not quite. So now we can look at the AlphaFold structure and we can see that they have good recall precision and F measure. And then if you look at the average DP score, so this is for the individual models, you can see that they are actually great. So they are a really good match to the NMR data. So I guess that means I don't have to solve NMR structures anymore. So they're always as good as the best individual structures or within like 0.01 from the best. So for example, here 0.81, 0.82. And the X-ray structures also match the NMR data pretty well in many of the cases. So what else can we learn? Oh, here's the conclusion. So yes, there's a very good correlation between the DP score and accuracy for NMR structures, and these AlphaFold structures fit the data very well. As the title of our paper said, they rival the accuracy of solution NMR structures. And also it's very useful for comparing structures solved by different refinement methods to see if that helps like improve the agreement with the NMR data. So like we did that with Rosetta, this paper in 2014 from Montelione lab, they was using Rosetta, and they found that it made the NMR structures look more like the X-ray structures so that they could be used, better used for molecular replacement. And so you can test like AlphaFold, like different methods, and see like which one matches your NMR data the best. Then we also have use a software called the Protein Structure Validation Software Suite. It's also being served in the Montelione lab at RPI, and you get a lot of structure validation, output scores. So these are knowledge-based structure validation scores. You get your alpha helices, you get violations like of your restraints, like per model. But then I wanted to focus on these global quality scores. So ProCheck phi psi is backbone, and ProCheck all is like all backbone and sidechain. And these are quality scores that kind of look at the stereochemical quality of the dihedral angles of the protein and like and see how they match, how good they are. And so Molprobity Clash Score is the software from the Richardson lab, and it looks at how many high energy like close contacts do you have in a structure. And so you get these raw scores, and then we have these normalized Z scores. So we took they're from a database of like, I think 250 high resolution X-ray structures, and normalized so that if it's at 0, that means it's at the mean. And so if it's a positive, +1 would be one standard deviation better than average. So having positive Z scores means these are better than average for high resolution crystal structures. And these are, NMR structures. So these are very good numbers. So we wanted to look at these structure validation scores for all the different models, because even though like it's pretty much known that like having good structure validation scores doesn't mean you have the accurate structure, because we know you could have like perfect validation scores and an inaccurate structure. You also want to match your data really well, and you want good quality validation scores. Roberto Tejero has been working on this software recently. He has a new version, PSVS 2, and that version will also do RDC analysis. So if you give it the RDC data and give you quality factor numbers. And so that's actually a very nice feature. Thank you, Roberto. So what were the validation scores? So we found that the AlphaFold structures like here and here had excellent quality scores, as we would expect. And they have excellent Z-scores. And in fact, in these cases shown here, the Molprobity clash scores came back with like NA, like it had 0 clashes. So they were basically perfect. And so we put that as greater than 3.5 standard deviations away from average for the Z-score. So these structures are very good quality, and they also match the data very well. So that's a win-win, right? That's our goal, is to have structures that match the data and also have good quality scores. So then we also decided to compare with the NH RDCs. And so, so thanks to Angus for talking about this. So basically we had only NH RDCs for 2 sets of each for 3 of the NMR X-ray pairs. So what do we have? He talked about how you can record a proton nitrogen HSQC without decoupling, and you can get this proton nitrogen J coupling. Then if you put the sample into some kind of alignment media and you get a little bit of residual alignment in the magnetic field, you get this additional term, which is the residual dipolar coupling measured. We actually didn't measure it this way. For those NMR X-ray pairs, the RDC data was collected in Jim Prestegard's lab, and he used similar to an RDC, but not 3D. He used like a JMOD, an intensity-based experiment. So we have these measured RDCs, and then you need to calculate the RDCs for model structures, and that was done using the single value decomposition that was also talked about by Angus, that that Roberto put into his software that's called PDB Stat. And so what you can see, so basically what we have here, we have NH RDC data. So independent from nosy data, we have orientation information about these NH bond vectors with respect to the magnetic field. So first you have to find like a global alignment tensor for your structure, like how does it align, and assuming like it's rigid and it's aligning in the magnetic field. Then you have information about how each NH bond vector that has an RDC, how each of them is the angle with respect to the magnetic field. So this is like independent of nosy data. You have validation NMR data that can be used for validation. So what is shown here is the sequence and then the RDC data. And so here we have the measured data in orange and then the calculated in blue, and then you get a quality factor, which is the the agreement of the how well it agrees with the data. So this is the equation that Angus was talking about, Q2. Some people use Q1. They're very similar, that if you have a lot of RDCs and you're in a lot of different angles that are sampled, but we decided to put both. If you have a low Q factor, like 0.27, that's very good. And so we can now use this to look at the AlphaFold structures. And so we did that. In the cases we had 3 proteins and 2 sets of RDCs, 2 sets of alignment media each, so polyethylene glycol and phage, and polyethylene glycol and a polyacrylamide gel. I believe this was like a stretched gel. So what we did is, so the lowest Q score is going to be in bold. So so lower Q score is a better agreement with RDCs. So in all 6 cases, the AlphaFold models had better agreement with RDCs than the NMR structures that were not refined with RDCs. So for example, 0.43, not refined with RDCs, and then AlphaFold 0.25. 0.25, and then AlphaFold 0.235, 0.47. Oops. and 0.5 and 0.37. So that was amazing, because that means that it's independently verifying the accuracy of the AlphaFold structures. Of course, when there's an asterisk here and here, it means that these NMR structures were refined with the RDCs. So then you would expect it to have better agreement with the RDCs. Kind of circular logic, but you use them, and so therefore you get better agreement. And then we also can compare to the X-ray structure, the RDCs, and see that sometimes the AlphaFold structure is even better agreement than the X-ray structure. So the AlphaFold Q-scores are actually close to those of the X-ray structures. So this was really interesting to us, and independent verification that these AlphaFold structures are very accurate. In this case, though, when we have RpR324 with polyethylene glycol and phage as a different alignment media, we found that, again, the AlphaFold had a lower Q-score than the NMR not refined with RDCs. We have 0.27, 0.45. But what is going on with these X-ray structures? Why are they higher? They had the worst agreement with RDCs. So that was a question that we wanted to look into further. So it turned out that when you look at the structural agreement, the AlphaFold structure and the NMR structure refined with NH RDCs has the best agreement. The GDT score of 99 is very good, and also the RMSE of 0.59. But we also see that the X-ray structure here is 86, and the RMSD between the X-ray and AlphaFold is 1.74. So it turns out that the X-ray structure has some slight differences in the structure. So this protein is a specialized acyl carrier protein that is believed that its biological function is to carry these long acyl chains attached covalently to a serine group and a phosphopantothioate group, and it has to open up this hydrophobic cavity to let the acyl chain in there. So in the crystal structure, there is a dimer interface that has buried surface area, and it makes this cavity. So it turns out that in the X-ray structure, compared to like if you you now have a slightly bigger hydrophobic cavity than if you had the coordinates that were solved by NMR or AlphaFold. So we wanted to look into this a little bit more to see if the NMR data could give us any idea of what was going on with this. So oh yeah, I also wanted to see if AlphaFold dimer could match the X-ray structure. So that was like a really interesting question that we had. So AlphaFold dimer is also super easy to run. You give it the sequence, you tell it's a dimer, and it came up with the almost exact same dimer interface as the X-ray structure, which was very satisfying. But if you look at it really carefully, you'll see there's actually slightly different orientation here. And so when you overlay just the monomer and look at it, you can see that the X-ray structure shown in gray still has that movement of that helix that opens up the cavity. And the AlphaFold dimer is still predicting basically the exact same thing that we got with AlphaFold monomer calculation. So they're giving basically the same structure. So the X-ray structure truly is having some differences. And if you look at global scores, you might not catch it, but if you look specifically at how can we look specifically to see like where these differences are coming from. So the thing we developed is called this double recall violation analysis. So what it's doing is it takes your peak list from your NOSI data, and you look at peaks that can correspond to an NOE that is satisfied by the NMR structure only, but then not by the X-ray structure, for example. So if you have two different structures, and in this case, we're using the medoid of the NMR structure, so just one. If you have two structures, the NMR and X-ray, you can now take your NOSI peak list and you can say which peaks are being satisfied by the NMR structure only, but not by the X-ray structure, and also vice versa, any structures that are satisfied by the X-ray structure only and not by the NMR. And then this was then plotted onto the X-ray structure. So this this residue 28 is the one that is satisfied the NMR structure only and not by the X-ray. And you can then if you can then do that with AlphaFold structure and the X-ray structure, and you can see that this is very similar to this because the NMR and the AlphaFold structure were very similar. And then if you also do this analysis with NMR and AlphaFold structure and you compare, you actually get that there's very few. So this is a count, like the number of violations. And so you can see that these structures are very much in agreement. So what does this mean, and how can we interpret it? So I thought this was excellent. This is very interesting. So it turns out that looking more closely at histidine 28, comparing between the AlphaFold, which is shown in yellow and also the gray, and the X-ray structure, which is blue here, we find that these peaks that are not being assigned by the X-ray structure belong to histidine 28. And so I couldn't go through like a talk without showing NOSI data. So here's NOSI spectrum from the aromatic region showing the histidine 28 H epsilon. And here's a peak that is for the T71 methyl group, which is right here. So it turns out that in the AlphaFold structure and the NMR structure, there's a short distance here that could be a hydrogen bond between the nitrogen of the histidine and the OH of 371. And then there's also NOEs or peaks between this 371 and this valine 64, which is in this alpha 3 helix. So by moving this histidine away, maybe it's we actually think the X-ray structure was at slightly lower pH of 5.5. It may be getting some protonated. You lose this hydrogen bond, and now it's like 7 angstroms away, and that then can open up this hydrophobic cavity. And so this may be very important for the function of this protein. So this was a very cool result that can be used to like learn biological information, or at least start to track down biological function of the protein forming an open and closed state. Here's that same double recall violation plot, and then we also plotted it as a 2D plot. So this is going to be the H1 violations, so from the basically the donor, so, and then also the indirect proton match. And so here we have what we see here is residue 28, and then these are the peaks that could be in orange that were satisfied by the AlphaFold structure only, but then not by the X-ray structure. There's a whole bunch of them here. And so these 2D plots are something we're exploring to see how useful they can be for finding structural differences between and matches to the peak list and the NOSI data. So I said I was going to come back to this structure, this model, and what we can learn. So it turns out this is the one where the AlphaFold structures had the lower DP score and not as good agreement with the NMR structure. So the first thing we did was we used the precision violations and plot those onto the structure. And you can see that there's, in red are the very bad precision violations. So means that based on this AlphaFold structure, you would expect short distances between protons in this helix that are missing from the data. And so that's why it's in red. So it turns out that that these evidence for this C-terminal helix that was predicted by AlphaFold 2 was not seen in NMR data. So then we did the double recall violation analysis, and we found peaks that were satisfied by the NMR structure only and peaks that are satisfied by the AlphaFold 2 structure only. And it turns out in this N-terminal helix, which in the NMR structure is in the middle of all the other helices, right here, alpha 1, this was not seen in the AlphaFold structure. And it has NOE peaks that are satisfied only by the NMR structure. Then also this C-terminal helix, which was not seen in the NMR structure, there's a few peaks here that can be satisfied by the AlphaFold 2 structure only. So it sort of suggests that the AlphaFold structure may be identifying one of a possible multiple conformation that is present in solution. And so we think that using AlphaFold may be and seeing these differences could actually be suggesting that we have multiple conformations going on in solution. And that is why this AlphaFold structure is not matching the NMR data as good as the NMR structure here. So conclusions. These AlphaFold structures are great. My work is done. We validated them with, we validated these models with NMR data. And then we also found, surprisingly, that the AlphaFold models match the NMR structure and data even better than the X-ray structure, despite the fact that AlphaFold was trained on an X-ray structured database, which was, I think, very surprising result. We found that this double recall violation analysis can be used to find localized structural differences if based on agreement with NMR data. So you can see in specific regions of the protein which one matches the NMR data better. We also found that in the case of a target that did have actually the NMR line widths and the heteronuclear NOE data actually suggested a lot of dynamics for this protein. In the case of multiple conformational states, that AlphaFold may be useful for identifying some of the multiple conformations present in solution, and that and so AlphaFold really did a great job of predicting structures in the case where there wasn't any conformational dynamics. So that was our conclusion. I'd like to thank the people in the Montelione lab. Guy, I realized you didn't have a picture here, so I added one. Here at RPI, we had, and Roberto in Spain, and a lot of the people that worked on the NMR structures in other labs, including Toshio Yamazaki in Japan.
This leads into the discussion about AlphaFold and NMR and what AlphaFold means for NMR, or how we could take advantage of this new tool. This is actually from a summary of history of CASP, and it starts with CASP. Start with CASP 14 in 2020. So these open circles are about 100 targets, for which structures were provided by, that were blind structures, which weren't available, and we're plotting GDT score versus the difficulty of the target. So difficulty means, can you find a template? Can you find a template for homology modeling? And what the open circles show is that, and those are the results from AlphaFold open circles. And what the open circles show is that most of the targets came in with GDT greater than about 80%. 80% means about 1.5 angstroms RMSD. And many over 90%. The average was about 90%. That's under 1 angstrom RMSD. And then also what you see here, the dashed line, that's the result for all participants in CASP in 2014. So you could see the difference. Well, I should say the solid line is a fit through those open circles. And the dashed line is for all participants except the AlphaFold. And then the other lines are the fits through the best results over the previous years. So you can see the great improvement. And what's important is that the AlphaFold predictions work well, even with the most difficult targets. And that's always been a challenge in CASP. And the other thing that's shown here are, as Nick talked about, the 3 NMR structures, T1027, T1055, and T1029, which came out with a very poor GDT score initially, but based on interactions with this group, we recognized there were problems with their data, and they redetermined their structure, T1029 revised with the data, and you see it's actually now, that it actually matches the AlphaFold prediction quite well. See, a point that hasn't been made is that the quality of the side chains is really outstanding, is really quite good. So this is a comparison between core side chains of an AlphaFold 2 model and the NMR structure T1055. And going back to the NMR X-ray pairs that Theresa talked about, so this is a plot looking at Chi-1 values or binned Chi-1 values for buried side chains. For either the crystal structure, there's NMR structure with Rosetta refinement, or the AlphaFold structure. And we're comparing against the crystal structure. And we see is that the agreement of the Chi-1 rotamer states is pretty good for these NMR structures, but it's even much better for the AlphaFold structures. The general conclusion is that for Chi-1 and Chi-2, the buried rotamer states have Chi-1 and Chi-2 values that are as accurate or better than those of the experimentally determined NMR structures. So how can AlphaFold models guide NMR data analysis? Well, we can use the prediction model to guide experimental data analysis. We can think about inverse structure determination, where now we have a hypothesis of the structure, and can we advance that? Can we use that structure to streamline the data analysis? And then can we use it for modeling multiple conformational states and energy landscapes? So, so this is one of the targets that Theresa talked about. And this is a target, Cas1055, which was very accurately predicted. The RMSD was under an angstrom for the backbone, and GDT score about 90%. And here's a summary of these statistics she described. So if we look at the AlphaFold model, or the NMR model for that matter, we find a lot of precision violations. These means there are short distances that are not explained by the no-see peak list. And when we worked with the person who did the structure, we learned that they made a very, very stringent peak picking, a very stringent threshold. They wanted to be very precise, but by being so precise, there isn't a good match between the chemical shifts in the no-see and the chemical shift assignments. And so no-see peaks are excluded from the peak list. So you have much fewer no-see peaks in the peak list than really count. And that comes up by looking at the model and looking at the short distances that are not consistent with the nose. So this gives a way to improve the no-see peak picking. Generally speaking, we need AI-based methods for peak picking no-see data to remove user bias from the structure and validation. So case 2, inverse structure determination. So this is actually going to become much more common. So this was the structure Nick talked about, Theresa talked about, that was but basically wrong. The GDT quality was low compared to the AlphaFold structure, but more important, the DP score was very low. And so what we did here was use the recall analysis to identify that there were severe problems in the no-see peak list. And we encouraged the experimentalists to go back and reanalyze carefully their no-see peak list. So we put a lot of encouragement on them to do that. They came up with a much better no-see peak list. And now they could increase, we could actually increase the quality of this data and redetermine the protein structure and improve the DP score. So this is the original structure mapping to the AlphaFold structure. And here's the correlation between DP and GDT. And you see it's not even correlated very well because we're not using a correct structure as the reference. But when they repicked the peak list and redetermined the structure, now you see a very strong correlation between GP and GDT. And the resulting structure matches the AlphaFold structure, and it matches the data much better. And kind of as an independent validation, we looked at the RDC plot. So these are the Q scores that we're talking about. And the original structure had acceptable Q scores. They were pretty happy with their Q scores for NH, and it's 3 different RDCs. The refined structure actually has somewhat better Q scores, and it actually calls into question using the RDC Q score for structure validation, because even some incorrect structures can have okay Q scores. And so this is really a kind of important example. And then finally is the case that Theresa talked about. In this case, the GDT between the AlphaFold structure and the NMR structure is quite low, but the NMR structure fits the data much better than AlphaFold structure. But when we look very carefully and we look at these precision violations that Theresa talked about, we find that there's a whole bunch of precision NOEs that can only be explained by the NMR structure, but not by the AlphaFold 2 structure. But on the other hand, there are some NOEs that can't be explained, the orange ones here. They can't be explained by the NMR structure. They can only be explained by the AlphaFold 2 structure. It actually appears as though there's a minor population of the AlphaFold 2 structure present in the solution, which are being represented by these NOEs that shown in orange. So the NMR data is best fit to the NMR structure. However, some NOEs are not consistent with the NMR model, but are in fact consistent with the AlphaFold 2 model. So it appears that AlphaFold 2 may be providing information about the alternative conformation in this case. And we think that's actually quite exciting. And it actually relates to some very nice recent papers. So as was explained in the first talk, a very important part of the AlphaFold pipeline is this embedding process where evolutionary covariance constraints and perhaps homology information is used to get some prior information that then goes into the main machine learning network. But what people have observed is if they use much shallower or even no multiple sequence alignments, that is, they don't use this information, then they get a broader distribution of conformations predicted. And when you have multiple states, you find the multiple states among that broader set of conformations. So this is actually a very exciting future direction about modeling multi-state structures with AlphaFold and potentially bringing NMR data into the story. So, AlphaFold and NMR. AlphaFold models do not replace experimental structures, but they bring us to a much higher starting point for our experimental studies. They can be used for guiding construct design, guiding NMR data analysis with AlphaFold models, like I just described with the problem with all those precision violations, validating models with sparse NMR data. For example, as we heard about with the SARS coronavirus MPRO, where RDCs and some NOEs are used to validate the models, guiding dynamic studies by providing models of alternative conformations, and of course, guiding protein interaction studies like ligand binding and biomolecular binding. So I think the point of this is that, and to lead into the discussion, is that AlphaFold doesn't replace experimental work, it enhances experimental work. And I think that point was also made earlier. So, and it's AlphaFold, of course, is not only relevant for NMR, but broadly for structural biology. The AlphaFold models can be used for guiding resonance assignments and model validation, some work we're doing. We're taking SACS data where we couldn't get experimental structures any other way and seeing if we could use the models and fit them into the SACS data. There's an project for modeling across protein families and looking at structure-based evolution. There was an idea in structural genomics that structure gives clues to function. Well, now we can go after whole proteomes and try to discover biochemical functions of proteins that way. We have the ability with AlphaFold to model multiple conformational states, so-called invisible states or alternative states of proteins. And we heard a little bit about modeling protein-protein complexes and protein-peptide complexes. This is actually quite successful. We've had good results. We're taking disordered peptides and modeling the complexes that they form when they bind to proteins. And then a very exciting idea, which was also spoken about earlier, is the idea of generative adversarial networks. This is an approach in which you try to make the image that you desire. And what people are starting to do is to make an image which is a protein design that is desired, and then use modeling, making many, many different models with the AlphaFold-like approach and assessing those models to be able to hallucinate a novel protein structure. In any case, I think this probably is a good point for our discussion, and I went a little long, so I'll stop here and let's have some discussion from the audience.